Hi, this is Guy Haskin-Fernald, and this video lecture is about chemoinformatics and pharmacogenomics. Now, chemoinformatics, or cheminformatics as it's also known, is really just the use of informatics and computation to study chemistry. And in particular, I'm going to talk about some aspects of it which are of importance for the study of pharmacogenomics. So we'll talk about the representation of chemicals in 2 dimensions and 3 dimensions. We'll talk about virtual screening, virtual libraries, and how they can be used for similarity searching, docking, and quantitative structure activity relationships. So to start with the representation, so you can't really study chemicals on a computer unless you can represent them. So that's the first problem we need to solve. And chemicals are real physical things. They're 3-dimensional. They have mass, charge, hydrogen donors, acceptors, etc. But computers, as you all know, really just store things as ones and zeros. So we have this problem of representing it on a computer. Just like we represent DNA with ACGT sequences, we need to find a way to represent chemicals. And so, for example, here's a picture of aspirin, acetylsalicylic acid, and we want to figure out how to manipulate that on a computer. Well, a simple way to do that in 2 dimensions is to use a graph. So you can take your chemical, and given what you know about the chemistry, you know that certain atoms are bonded to each other, and you can just draw those connections. And in this representation, the carbons are not labeled, but they are all the vertices without labels, right, such as these in the ring. The other atoms are labeled, and then you have 1 line for a single bond and 2 lines, like here, for a double bond. You can then use graph algorithms to compare your chemicals to each other, such as subgraph isomorphism. Now, in order to represent them and perform these computations, you're going to need to label all those vertices so you know which one is referring to which atom refers to which vertex, and then you can represent the connections between them using a matrix. And in this case, you put all of the atoms along the vertices and all along the columns, and if they have a connection between them, you give them a 1. So a 1 in this case means a single bond, and a 2 means a double bond. If there's zeros, that means there's no connection between them at all. And then using this representation, you can implement many interesting algorithms. If you want to take this a step further and represent it in 3 dimensions, you need the 3-dimensional coordinates. And in many cases, you can get those, and you see them as x, y, z coordinates listed over here. So now you can couple that with your labeling of the vertices and the connectivity between them, and you can use that to do complex algorithms for dealing with chemical and physical interactions now that you know where they're all sitting in 3-dimensional space. Now, a big innovation in cheminformatics came with linear notations, because this allowed you to represent very efficiently lots of structures of chemicals. And it's a linear notation where you essentially take that graph and you put it into an ASCII string. The most famous of these, or most commonly used, is the SMILES format, the Simplified Molecular Input Line Entry Specification. And in this case, you use your ASCII characters to represent the different atoms. You can use, if they're just next to each other, it's a single bond. You can use an equal sign, for example, for a double bond, and there's many other ways to represent aspects of chemicals. But then you need to figure out how to walk through the graph. Now, there's a few different ways where you choose your starting point, for example, by running hash algorithms to determine which atom is going to be your starting point. But the most important part is figuring out where to cut them. And so here you see these 3 lines showing where you're going to make a cut in your trajectory through the graph. So we're starting, in this case, with this oxygen, and you can see we have a double bond to a carbon, and then we make a cut, that's signified by this slash here. And that's represented in the SMILES code by the open parentheses. So you make that cut, and you remember that you'll have to come back here later, and then you continue to traverse your chemical until you have to make another cut over here. And you'll notice that when you enter the ring, you go from a capital C to a lowercase c, so lowercase letters are always in ring structures. And then finally you come back around and you unwind each of your parentheses, you make your 3rd cut here, and you're done. And this is a very efficient way. There are canonical representations, and you can store large quantities of these in databases. Another representation is a pharmacophore, and this is an interesting concept because you're associating your chemical with an important property, a three-dimensional property. So suppose you have several chemicals which all are known to bind the same protein. And in this picture, you can see we have this green structure here is representing the protein surface. And you can see the atoms labeled in there, and there's some angstrom distances between them, between 2 and 5, between these two, 7 to 8, between these over here, and 7 to 8 between these over here. And what you've done is you find a common three-dimensional pattern in all the molecules which are known to bind that protein in that particular site. And if you can find a set of them and find this 3D pattern, you store that and you call that a 3D pharmacophore, and you can have a 3 or 4 point pharmacophore. In this case, you see we have a 3-point with a triangle. There are other representations for chemicals, such as the IUPAC representation, where you see these strings, for example, and the international chemical identifiers represented by here, and there's other things called Marcouche representations, which are mostly used for patents. So you may have a chemical and you'll make a patent saying we have these ring structures together, and then we have these groups R1, R2, R3, and the document will go on to say, you know, these are the types of groups that may be associated with R1 and R2 and R3. So moving on, we can now represent these chemicals. We can use them to do some interesting things, such as virtual screening. And what virtual screening has done is finding similar molecules, or maybe finding dissimilar molecules. So it would be known as similarity searching or diversity searching. So if you have a chemical of interest, and let's suppose we're using like a SMILES code to represent them, you may say, I want to find other molecules which are similar to this. So you make a screen, represented here in blue, and you compare your molecules to that molecule of interest up here, and if they're similar enough, you save them and you bring them across. So you can essentially quickly scan a large database of chemicals saying these ones are similar to my molecule of interest, and I might be interested in this because I know that molecule is active with a particular protein, and I want to find all the molecules which may also be active, and then perform some additional searching and testing with those. So one way to do that would be to use graph methods using the 2D or even 3D structures that I showed you in previous slides, but they can be very expensive if you have a large database. For example, the most common substructure algorithm for graphs is an NP-complete problem. So you could basically use up all your time just trying to run the algorithms. So a way to avoid this is to use fingerprints, where you take a chemical and you simplify it even further from the SMILES code or the graph representation into a hash, where you essentially choose a bunch of features such as the presence or absence of this NH2, and you make it a 1 if it's there or 0 if it's not. If you can come up with a whole bunch of these hash codes, then you can determine how similar things are. One such system is called MAX, structural keys, and so these essentially are saying, do they have certain features? Some examples are, the number of hydrogens. Do they have a certain number of oxygens in them? Is there a ring? Does it have a heavy atom? You take each of these keys, you give them a number, and then you can describe a chemical with a set of these numbers. And in this case, you're looking at two chemicals above, and they have a lot of these numbers in common. So they will, in fact, you can then compare them using Tanimoto similarity. So if you take those two sets from above, you call them max 1 up here and max 2 down here, you can see there's a lot that are in common, right? 36 is in both, right there, 59 is in both, 74, 75. They have many, many in common. There are some that are not in both as well. So let's see, like 111, well, 111 is in both, but 107 is not in both, for example. So the Tanimoto similarity is defined as taking the intersection of the two and dividing it by the union of two. In this case, there are a total of 44 among both, but only 33 of them are in common to both sets, so you give it a Tanimoto similarity of 0.75. And it turns out that Tanimoto similarity is actually a pretty good way to compare chemicals. And if they have a very high Tanimoto similarity, there's a very high chance that they will share activities and both bind to the same protein, for example. However, it's not always that good. And here's a somewhat famous example where you're looking at morphine up here and codeine and heroin, which are all known to bind the same opioids receptors. And morphine and codeine have a 0.99 Tanimoto. With heroin, it's 0.95. But methadone, which is known to bind, and in fact is used therapeutically because of that binding, scores just a 0.2. And so by most measurements, you wouldn't suspect that a 0.2 Tanimoto similarity would imply that they're binding the same receptors. So you can't always rely on Tanimoto for these things. There are other molecular descriptors which are not often used for Tanimoto similarities, but they can be useful for looking at properties of the chemicals, such as the partition coefficient, which is the log of the ratio of your chemical in octanol versus the concentration in unionized water. The molecular refractivity, other topological indices and shape indices, and basically drugs which are good and can enter the body and get across membranes generally are within a well-defined range. And so there's certain rules of thumb. One is called Lipinski's Rule of 5, for example, which describes what a drug looks like in general if it's going to be able to get access to receptors. So I just before that I mentioned that the Tanimoto is not always predictive of a chemical binding the same receptor. So another approach to this is to use something called the dock algorithm, which is essentially taking a chemical and in 3 dimensions and trying to dock it into your receptor. So this requires having your 3-dimensional structure for both your protein and your chemical. And what you do is you fill these spheres up and you create a negative image of the binding site. So these spheres here are all generated to figure out how much space you have. You can then take your chemical and insert it into the binding site using these spheres, and you can also use physical and chemical properties of the groups surrounding the pocket with your what you know about the amino acids present there, and you can start to use physical and chemical simulation in order to determine if it binds. It is, of course, an expensive algorithm, so it's not going to be as fast as the other ones, but you gain some chemical and physical specificity that you wouldn't otherwise have. Now I'll just quickly talk about one last aspect of chemoinformatics, and that is quantitative structure-activity relationships. These are essentially just regression models that are used to predict some activity of a chemical. And the assumption here is that molecules, if they're similar and you describe them with these properties, that those properties will be predictive of some some other properties, such as an activity. And a famous example of this is predicting boiling point. So taking some features of chemicals, you can actually predict the boiling point very well. In the case of pharmacogenomics, we're interested in predicting things like effective concentrations of a drug and how does it bind to its receptors. And in this example with capsaicin, which is a compound you find in peppers, you can see that if you have a bunch of compounds that are known to be active and have different effective concentrations shown by these EC50s over here. So the compound numbers are given along here. You can see the effective concentrations. If you just look at the average, there's no relationship, but they are all active. And by going through a series of transformations and choosing the right molecular descriptors, you can actually find this regression line here where you do things like take the log of different concentrations and choose the right features such as the partition coefficients and other things that are describing how the molecule works. You can actually come up with a pretty good prediction given a model and determine how you may even be able to make a better molecule that may bind the receptor that you're particularly interested in. So that is the summary for chemoinformatics. We talked about representing them in 2 dimensions and 3 dimensions. We talked about virtual screening, similarity searching, and docking, and we finished up with a discussion of QSAR.